Somewhere around 7,000 years ago, a clay vessel sat in a stone room in what is now Georgia. Inside, grape juice. And something was happening that no one could explain. The liquid was moving, bubbling, hissing softly like something alive. The people who witnessed this did not know about microbes. They did not know about carbon dioxide, or glycolysis, or ATP. They simply knew that if they waited, something extraordinary happened. The Romans, centuries later, gave this process a name, from the Latin word fervor, to boil, because to them that is exactly what it looked like, a liquid boiling without heat, transformed by something invisible. And yet, the result was wine and bread and cheese. The result was civilization. We have been fermenting for over 10,000 years. Today, we begin to understand why. And in this series, we are going to take it all apart, together. Welcome to Good Gut Feeling. I'm so glad you're here. This is a space where we explore the magic of fermentation and what it means to truly nourish and listen to your body. Today we're going somewhere a little unexpected, and I think you're going to love it. This is the first episode of an 8-part series, The Science and Dynamics of Fermentation, a deep dive. Not just into recipes, but into the biology, the chemistry, the history, and the philosophy of what happens when you put the right ingredients in the right conditions and simply wait. Episode 1. This episode covers the foundations, the big picture, how fermentation works, who the microbial actors are, and where the boundary is between transformation and spoilage. Episode 2 takes us into the biochemistry, the energetics, the paradox of ATP, the invisible machinery that drives everything. Episode 3 introduces the full cast, the lactic acid bacteria, the yeasts, the acetic acid bacteria, the organisms that have shaped human civilization. Episode 4 is entirely dedicated to Koji, Aspergillus oryzae, one of the most remarkable organisms on the planet. You will not see fermentation the same way after this one. Episode 5 is about control, salt, pH, temperature, moisture, oxygen, the parameters that determine whether something becomes extraordinary or inedible. Episode 6 brings it into the body, the microbiome, the gut-brain axis, how fermented food affects the way we think and feel. Episode 7 is about safety, diagnosis, hygiene, risk, everything you need to know before you open a jar. And Episode 8 brings us to the future, bioeconomy, bioplastics, precision fermentation, cultural heritage. 8 episodes, one continuous question: what is actually happening in that jar? By the end of this episode, you will have a clear map of the entire field of fermentation science, who the players are, what they do, how they work, and why any of it matters. Whether you are new to fermentation or have been making kimchi for years, this is the foundation everything else builds on. Start here, and the rest of the series will fall into place. Let's start at the very beginning with a word. The word fermentation comes from the Latin fervor, to boil. And when you see a jar of active sourdough starter, or a bucket of fermenting cabbage releasing carbon dioxide, you understand immediately why the Romans chose that word. It looks like it is boiling. The liquid moves, it hisses, it rises, it breathes. And for millennia, that was all anyone knew. That it looked like boiling. That something invisible was at work. The science would come much later. The microscope was not invented until the late 1600s. Louis Pasteur would not definitively prove the role of microorganisms in fermentation until 1857. But the practice, the patient practice of putting food in a vessel, adding salt or thyme or both and waiting, that practice is at least 10,000 years old. Archaeological evidence traces fermentation to 7000 BC in China and Georgia. Wine, beer, fermented grains. Not as luxury, as survival. As the earliest known method of food preservation, and as something that, perhaps without knowing it, shaped human health, human culture, and ultimately human civilization itself. Let me take a moment to explain who we are actually working with, because once you know the players, every ferment becomes a story about relationships, not just chemistry. Three kingdoms of microorganisms dominate fermentation. They each have a different role, a different biochemical speciality, and a different relationship with the food they transform. The first and most important for most home ferments are the bacteria, specifically lactic acid bacteria. Genera like Lactobacillus and Pediococcus. These are the architects of acidity. They consume sugars and convert them into lactic acid, which lowers the pH of the environment, preserves the food, and creates that clean, bright, complex sourness that defines kimchi, sauerkraut, yogurt, sourdough. But bacteria do not work alone. Within a sourdough starter, something remarkable happens. The bacterium, Fructolactobacillus sanfranciscensis, and the yeast, Kazakhstania, Homilis, have evolved a kind of division of labor that is almost elegant. The bacterium consumes maltose, but not glucose. The yeast consumes glucose, but not maltose. There is no competition, there is only symbiosis. A shared table with no overlap. This is not accident. This is tens of thousands of years of co-evolution. The second kingdom, the yeasts, Saccharomyces cerevisiae. This single organism is arguably responsible for more human culture than any other species on Earth. Beer, wine, bread, whiskey, all built on the same enzymatic process. Sugar consumed, ethanol produced, carbon dioxide released. But beyond ethanol, yeasts produce dozens of secondary metabolites, esters, higher alcohols, aldehydes, that give fermented beverages their incredible aromatic complexity. And the third? The molds, Aspergillus oryzae, Koji, the national mold of Japan. This organism is in a category of its own, and we will dedicate an entire episode to it. For now, know this: Koji possesses an enzymatic arsenal of extraordinary depth. 65 endopeptidase genes, 69 exopeptidase genes, proteases, amylases, lipases, and the enzyme glutaminase, which converts glutamine into glutamic acid, the molecular source of umami. Koji does not just ferment, it transforms. And when these organisms come together in kombucha, in kefir, in sourdough, they form what scientists call a SCOBY, a symbiotic community of bacteria and yeast. Not just a culture, an ecosystem, a living community held together by chemistry and necessity. Now, let's look at what is actually happening at the molecular level. Imagine your kitchen, a jar of fermenting vegetables sitting on the counter. Inside that jar, millions of organisms, invisible, patient, purposeful, eating, producing, transforming. You cannot see them, but they are working right now. Here is the central biochemistry. Fermentation is, at its core, a question of energy. In the presence of oxygen, cells can extract 30 to 32 molecules of ATP from a single molecule of glucose. ATP, adenosine triphosphate, is the universal energy currency of life. That is cellular respiration, efficient, powerful. Fermentation, by comparison, yields just 2 ATP per glucose molecule. That sounds like a catastrophic inefficiency, and it is. But here is the paradox. This inefficiency is precisely what drives the production of everything we value in fermented food. Because to survive on only 2 ATP per glucose, a microorganism must process enormous quantities of substrate, vast amounts of sugar, creating equally vast amounts of metabolic byproducts. Lactic acid, ethanol, carbon dioxide, acetic acid, flavor compounds in numbers beyond counting. The inefficiency is the product. There is one more challenge the cell must solve. During glycolysis, the initial breakdown of glucose, the coenzyme NAD+ is reduced to NADH. If NADH accumulates, the entire process stops. The cell faces what scientists call metabolic paralysis. The solution is elegant. Pyruvate, the end product of glycolysis, acts as an internal electron acceptor. It accepts electrons from NADH, restoring NAD+, allowing glycolysis to continue. The result of this electron transfer is either lactate or ethanol, depending on the organism. This single biochemical loop, glycolysis, NADH accumulation, pyruvate reduction, NAD+ regeneration, is the heartbeat of every ferment you have ever made or tasted. Now, the question everyone asks at some point: how do you know if it has fermented or spoiled? This is not a mysterious question. It has a precise answer. The first and most important principle is pH, specifically the critical threshold of pH 4.6. Below this point, the environment becomes hostile to Clostridium botulinum, the organism responsible for botulism. The spores of Clostridium botulinum cannot germinate below pH 4.6. This is the absolute safety standard of lactofermentation. A correctly made sauerkraut or kimchi will reach this threshold within days. The lactic acid bacteria do this work for you, if you give them the right conditions. The second principle is salt. At a concentration of approximately 2% by weight for vegetable ferments, salt performs a remarkable act of microbial selection. It creates osmotic shock, drawing water out of unwanted bacteria, effectively dehydrating them. The lactic acid bacteria, which are salt-tolerant, survive and thrive. The pathogens do not. The third principle is temperature. The ideal range is between 18 and 22 degrees Celsius, that is 65 to 72 degrees Fahrenheit. Above 30 degrees, fermentation accelerates, but flavor degrades. The pectins in your vegetables break down too quickly. The result is mushy, unbalanced, often bitter. Below 15 degrees, fermentation slows significantly. Not dangerous, but patient. And then, there is the question of what you see on the surface. Cam yeast, a white, flat, matte film, is harmless. It is a yeast that feeds on the surface of the brine. Remove it and continue. Pathogenic mold is different. It is fluffy, hairy, raised. It carries color: green, black, pink, red. If you see this, particularly in the early days of fermentation before pH has dropped sufficiently, that batch is compromised. The diagnostic tool is simple: flat and white, fine; fuzzy and colored, not fine. And there is one final tool worth mentioning: backslopping. The practice of inoculating a new batch with a small amount of a successful previous ferment. This gives your new batch a head start, a ready-made community of lactic acid bacteria, an accelerated pH drop, and a stability that a fresh batch takes days to build. It is the oldest quality control method in human history. Fermentation is not just a food technique, it is a technology. and its applications have expanded in ways that would have been unimaginable to the Romans watching their grape juice bubble. Inside your gut, there are more microbial cells than there are human cells in your entire body. This community, the gut microbiome, is not a passenger. It is an active participant in your immune function, your metabolism, your neurochemistry. Research now suggests that somewhere between 70 and 80% of the immune system is housed in the gut, and that the bacteria living there produce approximately 90% of your body's serotonin. Fermented food supports this system in ways that are only beginning to be fully understood. It delivers living organisms. It delivers metabolites, short-chain fatty acids, vitamins, bioactive peptides that the gut uses directly. And through a process called external digestion, it pre-breaks down the very compounds that would otherwise be hardest for us to process: phytic acid, gluten, casein, complex starches. We will dedicate a full episode to this in episode 6. Beyond the body, fermentation is now a cornerstone of the industrial economy. Lactic acid produced by fermentation is the building block for PLA, polylactic acid, the most commercially significant bioplastic in the world. Aspergillus oryzae has been producing lipases for laundry detergents at industrial scale since 1988. And on the frontier of precision fermentation, researchers are now engineering organisms to produce proteins, vitamins, and materials directly from carbon dioxide and sunlight. C1 feedstocks. No soil, no agriculture. Just microbes doing what they do. And through all of this, through health and industry and biotechnology, there is the cultural dimension. Fermentation is one of the oldest shared human practices. The Kimyeong tradition in Korea, the Koji culture of Japan, recognized as national heritage, the sourdoughs carried by settlers crossing continents, fed daily, given names, passed down through generations. UNESCO recognized this dimension in 2013, when Japanese food culture, washoku, was inscribed as an intangible cultural heritage of humanity. Fermentation was not incidental to that recognition. It was central to it. Fermentation is, in the end, a relationship. Not a technique, not a recipe, a relationship. The human creates the conditions, the temperature, the salt, the vessel, the time, and the microbe in return does something we cannot do ourselves. It transforms raw ingredients into something with greater nutritional depth, greater flavor complexity, greater safety, and greater longevity than anything we could produce alone. This is what we call the domestication symbiosis, the co-creation of man and microbe, a partnership that is at least 10,000 years old, and one that is becoming, as we understand it more deeply, more relevant, not less. What starts as a jar of salted cabbage becomes, at the molecular level, a self-organizing ecosystem, an act of trust, a surrender to biological processes that are older than agriculture, older than language, perhaps older than fire. And that, I think, is worth understanding. Not just to make better kimchi, but to understand something fundamental about what it means to nourish yourself, about the intelligence that already exists in food and microbes in the long, patient arc of natural processes. That is what this series is about. And we are only just beginning. Today we covered the foundations: etymology, biology, biochemistry, safety, and the remarkable reach of fermentation as a technology. In episode 2, we go deeper into the biochemistry, the energetics of ATP, the paradox of metabolic inefficiency, and the invisible logic that drives everything you make in your kitchen. I hope this opens something for you. a question, a curiosity, a new way of looking at that jar on your shelf. Leave a comment below. What aspect of fermentation are you most curious to explore? I am building this series around what you want to understand. Thank you, from the bottom of my heart, for being here for this first episode. If this resonated with you, I'd be so grateful if you liked this video and subscribed to my channel. Every click helps this little community grow, and the more we grow, the more we can explore together. Thank you so much for being here. Until next time, take good care of yourself, inside and out.